So now let's see cell organelles. So first cell organelle which we will consider is mitochondria. As you might be knowing, mitochondria is the powerhouse of the cell and is important for generation of ATP. Well, mitochondria are round or oval in shape. Then their number varies from 100 to 300 depending on the requirements of that particular cell. So there may be cells which require a lot of energy. For example, parietal cells in stomach, right? They have to continuously produce hydrochloric acid, and for which they require a lot of energy. So the number of mitochondria in these cells is very high. Okay. These mitochondria are bounded by double membrane. So this is showing the outer membrane, okay, and this is the inner membrane of the mitochondria. So this outer membrane actually is permeable to water, and the inner membrane, you see, it is folded, right? It is thrown into folds, and these folds are known as cristae. And there are a lot of proteins which are embedded on these folds. So basically, any folds, wherever in physiology you see the folds, it increases the surface area of the membrane. So by being thrown into folds, lot of proteins can embed into this membrane. So it forms two compartments because of these two membranes. One is between the outer and inner mitochondrial membrane, and one is inside this inner mitochondrial membrane. So two aqueous compartments are formed, and the inner compartment is known as matrix. It contains DNA, ribosomes and enzymes which are required for replication. Yes, mitochondria also contains DNA. Okay. So what are the functions of mitochondria? Well, it is well known that mitochondria is the powerhouse of the cell. That means it is important for generation of energy, for which enzymes of oxidative phosphorylation are required. But apart from that, it also has a role in apoptosis. See, there is a protein cytochrome C which is present in between these two layers of mitochondria, and it has a role in apoptosis. Then the steps of beta oxidation of fatty acids also occur in mitochondria. Then it also regulates the level of intracellular calcium. Then final steps of synthesis of steroid hormones take place in mitochondria. Well, we will see later that steroid synthesis occurs in endoplasmic reticulum, smooth endoplasmic reticulum, but the final steps of synthesis occur in mitochondria. Also, it has a role in urea and heme synthesis pathways. Also, we said that mitochondria has DNA, isn't it? So, a zygote mitochondria is derived from ovum, right? And the inheritance is actually maternal. It is very important to know this, because mitochondrial inheritance has been used to track the evolutionary descent. Okay, plus the mitochondrial DNA has a very ineffective DNA repair system. Mitochondria has very ineffective DNA repair system, and it has a high rate of mutations. So nowadays so many diseases have been traced to mutations in mitochondrial DNA. Next organelle which we will consider is lysosomes. Lysosomes are vesicular organelles, and they contain granules with hydrolytic enzymes. And all these enzymes are basically acid hydrolases. That means they act in acidic environment. So, lysosomes have to maintain an acidic environment inside for these enzymes to be active. Well, this is brought about by hydrogen ATPases, which are integral membrane proteins present on the membrane. So these hydrogen ATPases act and bring in hydrogen ions inside the cell against their concentration gradient, and this maintains the acidic environment inside the cell for these enzymes to act. And this if you see, it's kind of protective also that these enzymes are acting in acidic environment, because if by chance these enzymes are released into the cytosol, then they will become inactive because the cytoplasmic pH is not that acidic. So, these enzymes are active only within the lysosomes. Now, there are special lysosomes in some cells, for example, WBCs. You know that neutrophils, basophils, eosinophils have certain granules, and these granules are basically these lysosomes. And then acrosomes in sperm, which is the cap which is present on the sperm, that is also a specialized lysosome. So, how do these lysosomes act? See, the primary lysosome is formed by pinching off from the Golgi apparatus, and it is containing the acid hydrolases. When this primary lysosome fuses with a phagosome, which is bringing in the waste material or toxic material from outside into the cell. So, this primary lysosome, when fused with the phagolysosome, forms the secondary lysosome. So, the digestion of this material is being done by secondary lysosome. Now, after digestion, some indigestible material remains, and that forms the residual body, and finally that indigestible material is excreted out of the cell by means of exocytosis. So, that is the way of functioning of lysosomes. Now, suppose if there are certain enzymes which are deficient in primary lysosome, then what will happen? Yes, in that case the lysosome will not be able to digest certain toxic materials, and there will be build-up of these toxic materials inside the cell. And these diseases are known as lysosome storage diseases, which are characterized by deficiency of lysosomal enzymes. Then this residual body, they need to be thrown out of the cell by exocytosis. But these residual bodies start accumulating in the cell with age, and they are considered as wear and tear pigment and can be seen in many cells with age. Also, they are implicated in certain pathologies also. So, pathological accumulation of residual bodies in the cell is seen in Alzheimer's disease. Then in the eye, it can be a cause of macular degeneration, and there are many other diseases in which there can be pathological accumulation of residual bodies. Okay, now let's talk about other organelle, that is ribosomes. Ribosomes are the protein factories of the cell, that is they synthesize the proteins, and there are of two types. One present on endoplasmic reticulum membrane. Actually, they are studded on the membrane of the endoplasmic reticulum, and then there are free ribosomes. These one on that present on endoplasmic reticulum membrane are responsible for synthesis of transmembrane proteins, then secreted proteins, and the proteins that are stored in lysosomes. On the other hand, the free ribosomes are responsible for synthesis of cytoplasmic proteins, that means the proteins which remain in the cell. These transmembrane proteins will go and attach on the membrane. Secreted proteins will go outside the cell, and the proteins stored in lysosomes will be present in that organelle lysosomes. But there are other proteins which are present in the cytoplasm itself, right? Those proteins are synthesized by free ribosomes. And there are proteins found in mitochondria and peroxisomes as well. So, these are all synthesized by free ribosomes. Okay, let's move to other organelle, that is the endoplasmic reticulum. Now, we considered that ribosomes were of two types, those which were on endoplasmic reticulum and those were free ribosomes. Similarly, endoplasmic reticulum is also of two types. One is rough endoplasmic reticulum and the other is a smooth endoplasmic reticulum. So, you see here in this diagram, this rough endoplasmic, here it is showing the rough endoplasmic reticulum, and it is in continuity with the nuclear membrane. This is the nucleus, this is surrounded by the nuclear membrane, and this rough endoplasmic reticulum is in continuity with the nuclear membrane. Then this is smooth endoplasmic reticulum. Okay, so you see there is a difference. This is this red. These are the ribosomes, and these smooth endoplasmic reticulum, they do not have rough ribosomes attached on their membrane. So what is this endoplasmic reticulum? These are connected sac-like and tubular structures, and these are studded with ribosomes. Now these rough endoplasmic reticulum are responsible for synthesis of proteins. They are responsible for initial folding of polypeptide chains, right? So because ribosomes are there and they are the ones which are synthesizing proteins, isn't it? So that's why it is responsible for protein synthesis, plus the folding of polypeptide chains which are forming is done in rough endoplasmic reticulum. Plus there is some modification going on when the protein is being synthesized. After translation of the protein, some modification goes on, that is addition of some side chains occurs, that is post translational modification, that is also done by rough endoplasmic reticulum. Then smooth endoplasmic reticulum, as we said that there is no ribosomes, and hence the term smooth, isn't it? So no ribosomes on a smooth endoplasmic reticulum. Then they are more tubular. We said that rough endoplasmic reticulum is both sac-like and tubular, but the smooth endoplasmic reticulum is more tubular. The functions of smooth endoplasmic reticulum include re-esterification of fats in enterocytes. See what happens, that fats which we take in diet, these are basically triglycerides, that is the fatty acids plus glycerol. They are absorbed separately as fatty acids and glycerol. That means they are first broken down in the lumen and then they are absorbed. Now after absorption in the enterocyte, again the fatty acids combine with the glycerol to form the triglycerides. So that is known as re-esterification of fats. Then smooth endoplasmic reticulum is also responsible for steroid synthesis. Then detoxification of chemicals occurs in smooth endoplasmic reticulum. Also it acts as a calcium store, that is the sarcoplasmic reticulum in skeletal muscle, right? So in skeletal muscle, that sarcoplasmic reticulum is acting as a calcium store.